In regions like Chablis and Sancerre, viticulture is defined by steep slopes and pale, heavily textured soils known as Kimmeridgian marls. These soils are composed of ancient Jurassic marine sediments. They contain up to 30% active lime and are packed with the fossilized shells of a tiny oyster called Exogyra virgula. That extreme density of calcium carbonate drives the soil pH up into highly alkaline territory. The alkalinity physically locks iron, a critical nutrient, into an insoluble form, preventing the vine's roots from absorbing it. Without accessible iron, the plant cannot synthesize chlorophyll. The leaves turn a sickly yellow and metabolic function stalls, a condition documented as lime-induced chlorosis. The precise limestone formations credited with producing some of the world's most precise white wines are, chemically speaking, toxic to the vines planted in them. The standard agricultural defense is mechanical. Growers graft European Vitis vinifera cuttings onto specific interspecific rootstocks bred to tolerate high lime concentrations. However, field performance remains highly inconsistent. Certain vines fail where they should thrive. The variability indicates that plant genetics alone do not fully dictate survival under alkaline stress. In 1996, researchers at the Catholic University in Italy published this paper investigating the interaction between vine rootstocks and rhizosphere microorganisms. They hypothesized that inoculating grapevine roots with specific microbial populations might artificially replicate the survival mechanisms of naturally lime-tolerant vines. The trial tested two distinct biological agents, an arbuscular mycorrhizal fungus called Glomus mosaei and a soil bacteria known as Pseudomonas fluorescens. Surviving calcareous soil requires a three-way biological negotiation. The vine and the hostile soil chemistry must be bridged by active microbial brokers. The researchers established a testing matrix using three rootstocks: 3309C, which is highly susceptible to lime, SO4, which is moderately tolerant, and 41B, which is highly tolerant. The naturally tolerant 41B rootstock survives through conservative energy management. It exhibits slow, highly controlled vegetative growth when placed under alkaline stress. By contrast, the susceptible 3309C rootstock expends immediate energy on rapid growth, rapidly depleting its resources and suffering severe chlorosis as a result. These three charts track physical shoot growth of each rootstock over 160 days. The top graph for the susceptible 3309C shows a steep upward climb in length. The bottom graph for the tolerant 41B charts a much slower, flatter trajectory. In the vineyard, the susceptible vine produces long, pale, sickly shoots, while the tolerant vine produces short, robust growth that manages its limited nutrient intake. Measuring physical shoot extension does not accurately reflect a vine's true health or metabolic function when operating under severe alkaline stress. To evaluate actual health, we must shift the analysis from external shoot length to internal metabolic capability, specifically tracking chlorophyll synthesis. Table 2 from the study isolates the chlorophyll content of the leaves across different samplings. Looking at the baseline failure of the untreated 3309C control group, the chlorophyll measurement drops precipitously to 1.06 by the third sampling. The vine is starving for iron. Yet, in that exact same susceptible rootstock, the Pseudomonas bacterial inoculation drove the chlorophyll concentration up to 1.99, a near doubling of the vine's metabolic capacity. Visually, the fatally yellowed iron-starved leaves physically regreened, pulling the vine back from the edge of physiological collapse. The presence of this specific bacteria completely negated the genetic vulnerability of the rootstock, actively restoring normal metabolic function in a highly hostile environment. How did microbes execute this recovery? The logical assumption is they stimulated a massive expansion of the physical root system to gather more nutrients. We test that assumption by examining the root dry weight measurements in Table 1. The data reveals a paradox. The VAM fungal inoculation actually reduced the physical root mass of the 3309C vines by 21% compared to the untreated control. This reduction in mass is a structural trade-off. Mycorrhizal fungi alter the root architecture, projecting a massive microscopic web of hyphae outward into the soil. The vine undergoes an efficiency shift. It ceases building bulky, energy-expensive root mass and instead relies on the external mycelial network. The standard root quickly exhausts the nutrients in its immediate depletion zone. The mycelial network can bypass localized alkaline blockages to scavenge phosphorus and trace minerals far beyond the physical root's natural reach. In extreme Kimmeridgian soils, brute biological force is a liability. Survival is dictated by the microscopic efficiency and reach of these fungal partnerships. While the VAM fungi expand reach and secure phosphorus, the specific threat of lime-induced chlorosis is iron starvation. This is where the Pseudomonas bacteria execute a highly specific biological intervention. The bacteria exude organic molecules, known as hydroxamate siderophores, directly into the surrounding soil. Siderophores act as targeted chelators. They bind directly to insoluble iron trapped within calcium carbonate, prying it loose and transporting it directly across the root membrane. This transport system completely bypasses the natural pH barrier, chemically hacking the limestone's restrictive properties. Returning to Table 1, we see how these two distinct micropopulations interact in the soil. The introduction of the Pseudomonas bacteria actively increased the vine's natural establishment of VAM fungi, raising the infection rate from 12 to 20.3%. The microbes do not passively survive the alkaline environment. They operate in a synergistic loop, dismantling the terroir's chemical lockouts and feeding the host vine. Vegetative survival is only the baseline metric. The ultimate goal of viticulture is sugar accumulation and fruit ripening. Table 1 records the final agricultural metric, fruit-soluble solids, measured in degrees Brix. The highly susceptible 3309C vine inoculated with Pseudomonas achieved a Brix level of 23.3. The baseline for the naturally tolerant SO4 control vine was also 23.3. The genetically unsuited vine achieved perfect parity in sugar accumulation with a rootstock explicitly bred for these conditions, entirely due to the bacterial intervention. Properly managed microbial mediation successfully overrides a genetic limitations of the rootstock, neutralizing the primary chemical threat of calcareous soils. Scaling up from the microscopic data to the world's most famous vineyards requires a recalibration of how we view the soil. The pervasive industry myth is that vines eat rocks, somehow absorbing minerality directly from the limestone bedrock. In reality, the vine's root system is largely blind to the geological bedrock. It only experiences the immediate chemistry of its localized rhizosphere. The expression of Kimmeridgian or Oxfordian soils in a finished wine is entirely mediated by the microbial brokers negotiating that extreme chemistry. To understand the origin of the world's greatest white wines, professionals must stop obsessing over the dead rock and start managing the invisible ecosystem that actively dissolves it.